The first is computational methods to simulate behaviors and compute mathematical models. The second is looking at biology to understand and deal with complex systems, which in this context is the biological concept of morphogenesis. It's a Latin word from the 19th century that originated out of biology to describe development and evolution of organisms. And the reason I've chosen to look into use the concept of morphogenesis is because part of my research is dealing with this idea of developing a science for the development and evolution of complex systems and design, in particular, city tissues, which is the third part, and that's dealing with the scale that I'm working at. And what drew me to enter this research is the fact that over 70% of the world's population will be living in cities really in less than half a century from now. And that's really remarkable if you think about it, because it took this long. I mean, recently, we just passed the 50% mark. And so this is really unprecedented if you think about it, and it has quite heavy consequences. And very little has changed since really the past century. Modernist ideologies tend to be dominant in most of the urban approaches today. The idea of kind of superimposing blocks within existing tissues as a solution to deal with density and expansion. Zoning is often used as kind of a top-down approach for organization, but it has a limited capacity for dynamic reorganization as populations and economies grow or decline, and environments and climate change. But slowly, we've started to change our perception as a result of that, and we're starting to approach cities not so much as top-down, but as entities that emerge from the bottom up and are in a state of continual flux. So they're continuing to become more and more complex with the addition of new sources of energy and information. We have unprecedented shifts in social patterns. They're continuing to increase in density with the continued migration of people from rural areas to cities. And in many ways, cities are complex systems composed of smaller subsidiary systems that work together. And to deal with this level of complexity, it's important to identify the dominant systems that drive city morphologies. And how we do that, the question that comes up basically is, how do we deal with this level of complexity? And to understand how to model complex systems, we must first define what complex systems entail. Complexity theory is a comprehensive subject. It spans across various disciplines, including mathematics, physics, computer science, and biology, each, among others, obviously. And each discipline has a slightly modified definition of complexity that's pertinent to its field of study. In general, though, complexity theory can be summed up as the theory of dealing with multiple systems whose interactions or relations lead to emergent results or behaviors on a collective level. And this is kind of what goes back to biology. The concept of complexity has recently started to enter the domain of architecture and design, but other fields such as biology, physics, and so on, they've been dealing with complexity theory and systems thinking for at least a century now or more. Processes from the natural sciences offer great potential in exploring and understanding complex systems. You can see the quote on the screen, where the physicist Yanir Baryam describes biological development as an approach to kind of dealing with or designing complex systems. So there are two ways in which concepts from biological development have influenced my research. through computation. The first is deals with the structure of data. In biology, I'm looking at the structure of the DNA and how genes carry data from one generation to the other, as well as how data is structured for biological morphogenesis. The second is sensibility or feedback within design models. And I'll explain each one in more detail. So here with data, I focus on the structure of data in living forms and using that in the computational domain to structure data and come up with an approach to design. And while recent advancements over the past, I would say, decade now, in computation and technological tools have allowed us to kind of deal with vast amounts of data, the design profession, includes architecture, is still lagging coping with this abundance of data. And being able to manipulate it has really proven challenging. Over the past half century, we've seen quite a significant amount of advances in the field of science and mathematics towards understanding complex systems. One of the most profound discoveries has been the structure of the DNA and storing biological data, giving us a greater understanding of the genome, in particular its role in determining the physical characteristics of an organism or its phenotype. The genome carries within it all data necessary for the formation of a given life form. Hence, many say it is the blueprint of a living organism or living life form. And our genomes are composed of strands of DNA that coil into what are called chromosomes. Different organisms have different numbers of chromosomes, but the genes in these chromosomes tend to be, they're basically very similar, identical. They're responsible for the different characteristics, but they share a large amount of similarities. So what do these instructions look like? If you were to look at DNA code, which makes up our genes, they're composed of 4 letters. The sequencing of these letters is what differentiates us from one another. This is what a typical DNA code could look like, a series of letters one after another. And what is fascinating is that all these instructions for the formation of organisms come from these 4 letters. That's simply the sequence of the letters that is responsible for the variation. And in the late 20th century, the Human Genome Project was a big step in a way, because it was able to open up the path to decoding the relationship between genomic code and how form is generated. It begins to inform us on what the sequences may really mean, and how data is transcribed. So if we were to diagram the relationship between these genes, which are in these chromosomes, it would look something like this. And what you're seeing in the circle in the middle are the 23 human chromosomes, which are here. And the 3 white ones are dog chromosomes. And the color that's coming out of them is beginning to link the similarities that exist in dog chromosomes to the similarities that exist in human genes in the chromosomes. In a way, it's remarkable because you're starting to see how much is the same throughout different genomes of different organisms. But they're able to, through their sequencing, differentiate us on such a vast scale. Now, hierarchy exists in genes. These genes act together on a higher level to produce what are referred to as Hox genes. Hox genes regulate, they're responsible for the formation of body parts and limbs. And again, among different species, we're able to see a striking similarity in the structure of the Hox genes and how they control the overall formation of an organism. And you're seeing, for example, in this diagram, the Hox genes of a human, the Hox genes of a Drosophila, and how they are so similar in controlling certain body parts. So why is all this important? Well, because we're beginning to understand how data is structured and managed in biological development, which in turn lets us understand how complex systems are controlled. And what we're looking for are reductionist models through which we can create simple data structures that manage and produce complex systems. If we want to simulate this type of behavior without structuring data, it would prove to be very difficult, especially in evolving systems. So bringing it now to design, one way is to start with very basic instructions. Since we as designers deal with geometry, we're going to start with very basic geometrical instructions. On the left, you can see the population. P1 stands for population 1. We're beginning to keep track of the individuals. On the right, we're beginning to give them the instructions and keep track of the genes that act on them. And one of the biggest challenges in constructing digital models is structuring that data in a way that can be both managed and manipulated. So this is a step really in beginning to track, manage, and manipulate data as we begin to evolve systems. This diagram begins to kind of show in further detail what the codes in the previous diagram look like. It's beginning to kind of construct now a gene, and we're able to choose from this and plug it into the previous slide. So if we can produce now genes and we're able to now start manipulating them and activating them, we can begin to make variations within design. Here I've introduced a gene that basically cuts a hole into a cube, which begins to change the typology of the object and from an architectural point of view, from, let's say, a block into what could be perceived as a courtyard opening. And in this image, on the right, we're beginning to see the genome of a particular object again, where on the bottom part of the diagram, you're beginning to see kind of how the Hox genes begin to act and control the individual genes. And what we're really able to do is to now keep track of these objects, because you can see now, for example, this belongs to P9, population 9. Keep track of them as they continue being developed. Now, the second important feature of complex systems is the implementation of feedback, allowing them to adapt to their environment. And when we look at that, data in itself is somewhat quite limited. Structuring data is one thing, but manipulating it and using it to evolve a model is another. So feedback plays a critical role in not only evolving a model, but allowing it to adapt to its environment. And feedback loops allow information to be transmitted between different members of the system, but also between a system or multiple systems, as we'll see. One example of biological development is cell differentiation. Feedback plays a critical role during this process. And while very little is known in the relationship of genes and their ability to generate biological form, many studies have pointed to the role of feedback between gene expression and cell differentiation, which determines the cell structure and function and is a fundamental aspect of morphogenesis. This branch of biology in particular is kind of categorized under gene regulatory network. But in other words, what we're beginning to see is that genes regulate morphogenesis in biological organisms through feedback. And in the early 1960s, researchers started to explore the role of evolution and how it could be implemented in the domain of computation, which is referred to as evolutionary algorithms. Perhaps the most well-recognized of this research is the work of John Holland in the field of genetic algorithms. And his work was kind of based on the Darwinian notion of evolution and adaptation, how living systems evolve or change in response to other organisms or to a changing environment, and how computer systems can use similar principles to be adaptive as well. Genetic algorithms use biological evolutionary concepts in design. For architects in particular, John Fraser's book on evolutionary architecture is really an extraordinary book for introducing this concept in the domain of architecture. But the logic, as can be seen in this diagram, is based on three fundamental principles, really. It's evaluation, based on fitness criteria, selection, based on the fittest individuals, and reproduction through breeding or self-replication. Now, we can use GAs to define the genes that control the morphology of digital objects. Breeding genomes will then lead to differentiated phenotypes. And sometimes, you can get something that's not expected, but then that's where your fitness criteria comes in. And we can use this structure to begin to make generations within a given criteria. And these generations are then ranked, and based on those rankings, we can then breed again for the fittest genes to continue getting closer to the design goal. And as the model becomes more complex, the top shows, okay, original gene, digital gene. The second row, we're beginning to introduce mutation. The third, we're beginning to have Hox genes, so we can begin to further refine what we're looking for, but at the same time, we're able to kind of get a variation that fits within each design criteria that we're seeking. We then rank the populations, and as you can see in this diagram, as the population continues to go up, each generation continues to get fitter and fitter. So this video is, and I would hate to be a Drosophila, just because there's everything they want to do is on a Drosophila, but it's looking at embryonic development of a Drosophila. And as the embryo develops, cells begin to differentiate from generic cells into specialized cells. And these specialized cells then begin to form a population of tissues, which kind of happens through this kind of feedback loop between the cells and understanding where their location is or what they're supposed to do. Cells that belong to a specific tissue type adhere to one another, but don't adhere to cells from a different tissue type. And again, what we're seeing now is the formation of architectural features such as the body axis and tissue layers from a very early stage, which are resultant of the genes. But at the same time now that the cells are differentiating, what we're beginning to see now is the formation of the tritial tubes, the network that kind of goes through the organism. It can begin to distribute energy and so on. So this shows now simultaneously as that's occurring that there's another process, another system that's actually forming as well. And so what we do now is we can begin to look at mathematical models and begin to incorporate them into the design process. We saw earlier the kind of formation of objects through evolutionary algorithms, and now we're beginning to see the formation of networks through mathematical models. This is very straightforward. We're beginning to kind of just use angles and recursion. And we can kind of use this idea of branching to generate kind of different geometrical outputs. But what we see in biology and what we see in biological development, I should say, and complex systems is this idea of the formation of multiple systems at the same time. So I now then questioned and said, okay, what if we have different networks that want to have different metabolic behaviors, but kind of develop simultaneously? What would that begin to do? And we begin to now incorporate that idea into one model and allow them to kind of co-develop, where they begin to kind of form a single tissue in a way. And if we want to bring in a system of a complete different type to somehow be integrated with that, we can also, I guess there's two slides in this one. This slide, let's see. Is it? I should probably take that slide out. Okay. There we go. And we start to introduce now this idea of kind of the cells working in a way developing with the network and this relationship. So at the moment, this is kind of the point that I've gotten to with the research and kind of introducing multiple systems and looking at how they can begin to inform one another and kind of bring in those concepts of evolutionary algorithms within a design model and begin to use feedback as a method of evolving it as a complex system that can then begin to really generate city tissues. Thank you very much.